and welcome to Spotlight on Antibodies, a podcast from Biointron, where we bring you the latest antibody industry trends. I'm your host, Jovina Solistio, and this month in May, top news includes BCMA-Targeted Therapies, Multifunctional Natural Killer Cell Engagers, and Engineering IgE Antibodies. First up, progress in BCMA-Targeted Therapies. Multiple myeloma is a hematologic malignancy characterized by the clonal proliferation of malignant plasma cells within the bone marrow, often resulting in anemia, bone lesions, hypercalcemia, and renal dysfunction. Despite recent therapeutic advances, multiple myeloma remains incurable, and most patients ultimately experience relapsed or refractory disease. A prominent therapeutic target is B-cell maturation antigen, also known as BCMA, a cell surface receptor expressed almost exclusively on plasma cells and malignant myeloma cells, making it a highly specific and clinically actionable target for immunotherapies. On April 28, 2025, the European Commission approved linvoseltamab, or linvoseltamab, for adults who have undergone at least 3 prior therapies, including a proteasome inhibitor, an immunomodulatory agent, and an anti-CD38 monoclonal antibody. Linvoseltamab is a bispecific antibody targeting BCMA and CD3, facilitating T-cell-mediated cytotoxicity against multiple myeloma cells. The response-adapted dosing regimen allowing for every 4-week administration post-VGPR offers a significant advancement in convenience and treatment sustainability. Meanwhile, belantamab mafodotin, or Blenrep, a BCMA-directed antibody-drug conjugate, has received UK MHRA approval for use in combination regimens. These include Blenrep with bortezomib and dexamethasone, or with pomalidomide and dexamethasone, following at least 1 prior line of therapy. The findings establish Blenrep's efficacy in combination therapy despite its earlier withdrawal as a monotherapy in the US due to the DREAMM-3 outcome. Ocular toxicity, a class-related effect, was effectively managed via dosing adjustments. Furthermore, SB2001, a trispecific antibody engaging BCMA, CD38, and CD3, has been granted Fast Track designation by the FDA for patients with relapsed refractory multiple myeloma who have received at least 3 prior lines of therapy. Engineered for enhanced tumor targeting and reduced off-tumor toxicity, ISB 2001 demonstrated an 83% ORR in dose levels 3 to 7 during a phase 1 trial, with a CR rate of 22% and VGPR of 50%. Responses were observed even in patients previously treated with CAR T cells or bispecific antibodies, indicating potential utility in heavily pretreated and refractory populations. No dose-limiting toxicities were observed, and all cytokine release syndrome events were grade 1 or 2. These collective findings reinforce BCMA as a central target in relapsed refractory multiple myeloma and demonstrate the growing sophistication of immunotherapeutic approaches. Approvals of new bispecific and ADC combinations, the clinical advancement of trispecific formats, and the incorporation of adjunctive therapies like IVIG prophylaxis reflect an expanding and increasingly personalized therapeutic armamentarium aimed at prolonging remission and improving survival outcomes in this challenging disease. Natural killer cell-based therapies are rapidly gaining traction as immunotherapies, offering advantages over traditional T-cell-based approaches, particularly in safety and specificity. While T-cell engagers, TCEs, have demonstrated clinical efficacy, their use is limited by toxicities such as cytokine release syndrome. In contrast, NK cells possess potent cytolytic capabilities with reduced pro-inflammatory cytokine production, making them ideal candidates for bispecific engager platforms. Recent studies have advanced our understanding of NK cell engager, NKCE, biology, and demonstrated their potential in treating diverse and refractory malignancies. Recent comparative analyses have positioned NKCEs as a promising alternative to TCEs, particularly due to their lower propensity to trigger CRS. A study published in Cell Reports highlighted the mechanistic differences in tumor necrosis factor production between NK and T cells, attributing NK cells' lower TNF secretion to reduced intracellular trafficking and processing. This foundational insight has enabled the design of NKCEs that co-engage activating receptors such as CD16 and NCR1, while incorporating interleukin-2 domains to sustain NK cell survival, proliferation, and cytotoxic function. These multifunctional NKCEs integrate features commonly associated with antibody therapeutics, such as engineered Fc regions for optimized binding and afucosylation to enhance antibody-dependent cellular cytotoxicity, or ADCC. Benchmarking studies showed that NKCEs could be configured to bypass the limitations of receptor downregulation and low NK cell proliferation, inducing sustained tumor cell killing with reduced cytokine release. As a new class of antibody-derived therapeutics, multifunctional NKCEs offer modularity, specificity, and reduced immunotoxicity, establishing them as strong contenders for next-generation cancer immunotherapies. Glioblastoma is a type of cancer that starts as a growth of cells in the brain or spinal cord. Traditional immunotherapies, including checkpoint inhibitors and T-cell engagers, have demonstrated minimal efficacy in this context. In response, researchers at Purdue University have developed a novel antibody-based NKCE that is designed to simultaneously improve NK cell trafficking to glioblastoma lesions and mediate high-affinity tethering to tumor cells. The construct utilizes antibody scaffolding to engage NK cells through engineered Fc domains while targeting glioblastoma antigens. Notably, in vivo murine models demonstrated extended survival and enhanced intratumoral NK cell infiltration following treatment. Importantly, the platform supports off-the-shelf administration and can be integrated with both autologous and allogeneic NK cell products. Meanwhile, in a phase 1 trial, AFM13, a bispecific antibody targeting CD30 and CD16A, was combined with cytokine pre-activated cord blood-derived NK cells to treat patients with CD30+ lymphomas refractory to brentuximab vedotin and anti-PD-1 therapies. The therapy demonstrated a favorable safety profile with no cytokine release syndrome, neurotoxicity, or graft versus host disease. NK cells peaked in circulation within a day, trafficked effectively to tumors, and persisted up to 3 weeks. Clinical outcomes were a 92.9% overall response rate and a 66.7% complete response rate, with several patients maintaining remission for over a year. Building on this momentum, a first-in-human trial has been launched for XB628, a bispecific NKCE targeting NKG2A and PDL1. Developed by Invenra and Exelixis, XB628 designed to overcome immune suppression in solid tumors by simultaneously blocking 2 inhibitory checkpoints. This dual targeting approach aims to enhance NK cell activity within the tumor microenvironment, reflecting the next wave of innovation in bispecific immunotherapies. Let's move on to the topic of engineering IgE antibodies to overcome tumor immune suppression. Recent studies highlight the leveraging of distinct immunological functions of IgE to augment or replace conventional IgG1-based therapies, particularly for treatment-resistant solid tumors. Building on growing evidence of IgE's pro-inflammatory tumor reprogramming capabilities, novel hybrid antibodies combining IgE and IgG1 effector functions, known as IgEGs, have been developed to simultaneously engage multiple immune pathways. These insights converge on the development, functional characterization, and therapeutic implications of IgEGs, as well as a deeper exploration into IgE-driven immune modulation within the tumor microenvironment. In a recent study led by researchers from King's College London and Epsilogen, hybrid antibodies were constructed using variable domains from trastuzumab and a novel anti-HER2 clone, yielding Tras-IgEG and 26-IgEG respectively. These constructs were shown to express efficiently in mammalian cells, retained high solubility and stability, and were amenable to purification via standard protein A chromatography. Critically, they preserved both Fc epsilon Ri and Fc gamma Ri binding capabilities, demonstrating robust dual isotype functionality. Their findings suggest that while IgEGs are functionally competent, their potential additive benefits in vivo may be masked by current model limitations. Another study described a new type of antibody which stimulates the immune system to target cancer cells, which slows tumor growth, according to new research. In HER2-expressing treatment-resistant breast cancer models, anti-HER2 IgE antibodies reduced tumor growth and elicited a shift from immunosuppressive to immunostimulatory environment. These changes were characterized by enhanced infiltration of cytotoxic CD8 plus T cells and decreased regulatory T cell TREG populations, reflecting a rebalanced effector to suppressor cell ratio. Importantly, these immunologic transformations occurred in tumor types unresponsive to conventional IgG1 treatments, underscoring the unique therapeutic value of IgE. Further mechanistic insights into IgE-driven TME modulation were provided by studies of MOV18 IgE in ovarian cancer. Patient-derived macrophages, which typically exhibit immunosuppressive phenotypes and support TREG expansion, underwent robust pro-inflammatory repolarization upon MOV18 engagement. This reprogramming limited TREG-promoting interactions and enabled the expansion of CD8 plus T cells, an immune signature associated with favorable prognosis. RNA-seq and flow cytometry analyses confirmed emergence of a hyperinflammatory macrophage subset. Evidence from patient biopsies in a phase 1 clinical trial supported these ex vivo findings, confirming increased CD68 plus macrophage and CD3 plus T cell infiltration post-treatment. Beyond immediate effector functions, IgE also exerts long-term regulatory effects on adaptive immunity. As reviewed in a recent publication, IgE can function as a physiological adjuvant, enhancing antigen-specific secondary responses. Regulatory mechanisms, including autoantibodies against IgE and post-translational modifications such as glycosylation, influence its immunogenicity and persistence. These underexplored features could inform next-generation designs of IgE-based or IgEG hybrid therapeutics aimed at sustained immune activation. Collectively, these findings illuminate the potential of IgE in oncology, both as a standalone therapeutic and as part of hybrid constructs like IgEG. However, the full utility of IgEGs remain constrained by preclinical models that inadequately reflect human immune complexity, particularly with respect to mast cell and macrophage representation. The development of improved humanized models and Fc-engineered variants may develop their synergistic capabilities. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow Biointron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.